Good afternoon, everyone. I'm Stephen Burley. I'm the director of the Institute for Quantitative Biomedicine and also the interim director of the Rutgers AI and Data Science Collaboratory. I'm delighted to welcome you here to today's Wednesday seminar. We're hosting Andrew Santiago-Frangos from UPenn. But before I start with his introduction, there are a few housekeeping announcements. Michael, if you will, please. I want to draw your attention, anybody working on large structures, either here in the room or online, should take note that Brenda Vallant and Monica Sakaran will be presenting an office hour on depositing integrative hybrid method structure data to the PDB archive. This is a relatively new departure for the PDB to be housing this information within the main archive, and bodes very well for the future of understanding how large macromolecular machines work. Next slide, please. We will have another office hour led by Juan Segura, who's part of the PDB team in San Diego. He is the sequence annotation viewer is his brainchild on rcsp.org, and he'll be providing instruction and advice on how best to use that to understand connections between the linear amino acid sequence and the 3D structure. Next slide, please. We rely on your feedback. All feedback is good, particularly constructive negative feedback. So please use that QR code on the bottom left to connect with our user survey and provide feedback and get entered into an opportunity to win a mug featuring the artwork of the famous David Goodsell, who authored and edited the Molecule of the Month series for 2.5 decades. Next slide, please. So bringing me to today's speaker, Andrew Santiago-Frangos from UPenn. Andrew is the M. Jane Williams and Valerie Vargo Presidential Assistant Professor of Biology at UPenn. His work bridges bioinformatics, biochemistry, and cryo-electron microscopy to study the mechanisms and applications of immune systems. Prior to coming to Penn, in January of 2024, he received a PhD at Johns Hopkins with Sarah Woodson and was a postdoctoral fellow at Montana State University with Blake Wiedenheft. He was a Simons Foundation Fellow of the Life Sciences Research Foundation, received a Burroughs Wellcome Fund PDEP Award, and is the recipient of a Mosaic K99 Postdoctoral Career Transition Award from the NIH and additional funding from NIGMS. He's a leading junior scholar in the field, having published 19 papers since 2015, and is co-inventor on a patent of a novel Cas-based RNA diagnostic. So, Andrew, it's great to have you here. Thank you for making the trip up from Philadelphia. The floor is yours.
Okay. Thank you so much for the very kind introduction, Stephen. And thank you so much for having me here to share some of my research with you. So today I'll tell you about a story that I conducted as a postdoc in Dr. Blake Wiedenheft's lab at Montana State University. And then I'll also transition into telling you a little bit about our future directions and our progress there. All right, so the world is awash with bacteriophages, such that bacteria are experiencing viral pandemics on a continuous basis. There are estimated to be about 10 to the power of 30 phage particles on Earth, which is a massive number to try and understand. So one way to visualize that is to visualize every grain of sand on this planet across all the deserts, all the beaches, underneath all of the oceans, and then multiply that visualization by 1 trillion. And that's how many phage are all around us. As you might imagine, these huge number of phage have a large ecological impact. Roughly 30% of the ocean's biomass is estimated to be released on a daily basis. And that process is critical for the cycling of carbon and nitrogen in our environment. Now, bacteria aren't passive in this war against phages. They've evolved hundreds of different antiviral defense systems. A few of them are shown here. A recent discovery in our field has been that many of these prokaryotic antiviral defense systems share an evolutionary ancestry with what we had previously considered to be components of eukaryotic innate immunity. CRISPR is special in two key ways from this statement. First, it is the only adaptive bacterial immune system that we know of. And second, so far no CRISPR variant has been found in eukaryotes that provides immunity at least. So this barrier of what we think of as prokaryotic versus eukaryotic antiviral defense systems is starting to disintegrate. Now, we have several esteemed CRISPR biologists here at Rutgers studying the very basic biology of how CRISPR systems provide immunity. But to make sure that everyone's on the same playing field, I wanted to start with a basic introduction to show how CRISPR provides adaptive immunity against phages in bacteria. CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. And that acronym describes this genetic architecture that was first discovered in 1987, where you have these repeated DNA motifs, shown as these R's and diamonds, that are interspersed with unique DNA sequences, shown as these colored boxes. The CRISPR-associated integrase, Cas12, grabs a fragment of viral DNA and integrates it at the first repeat in the CRISPR array, establishing this chronological record of past infections. And this makes a DNA-based memory of this phage infection that will then act as a molecular vaccination card. Next, this CRISPR locus is transcribed into a single piece of RNA that is processed into individual guide RNAs. The Cas gene operon is transcribed and translated into Cas proteins. And these Cas proteins assemble with the guide RNA to form what is called an effector complex, which surveys the cell looking for complementary nucleic acid sequences, either RNA or DNA, depending on the exact type of CRISPR system. And when it identifies that complementary nucleic acid sequence, it then signals its degradation through a variety of different mechanisms. If you've read about or used CRISPR-based gene editing or diagnostic devices, then you've likely been using enzymes involved in this last stage of immunity called interference, such as Cas9, Cas12, or Cas13. The pressure of phage predation has caused a diversification evolution of many diverse CRISPR systems, but they all provide immunity in pretty much that same three key step process. These different CRISPR systems can be broken down into two main classes that is based on whether they have multi-subunit complexes or single protein complexes. An example of a single protein complex would be Cas9. These different CRISPR systems target viral DNA, and some of them target viral RNA. Recent surveys have shown that there's actually a previously underappreciated diversity and number of RNA phages in the environment. And it seems that many of these CRISPR systems have evolved to adapt to and degrade viral RNA. Common to all of these CRISPR systems is a CRISPR locus that will produce the guide RNAs for the effector complexes and the adaptation machinery Cas12. And then sometimes added onto this adaptation machinery are ancillary proteins like Cas4 or reverse transcriptases that promote adaptation to either DNA, viral DNA or viral RNA. When I first started my postdoc in Blake's lab, I became interested in this first step of immunity called adaptation. And specifically how that process is regulated. So the exact example that I chose to study was in Pseudomonas aeruginosa, PA14, which is an opportunistic pathogen. This pathogen has a type 1F CRISPR system that's characterized by this special fusion of a Cas3 domain to the Cas2 subunit to produce this heterohexameric complex called Cas123. This Cas123 integrase is very specific. It will grab a fragment of viral DNA and integrate it at the CRISPR repeat sequence. But as I mentioned earlier, it somehow ignores most of the repeats present in the genome, of which there are 30 in this organism, and specifically integrates new spacers at the first repeat in a given locus. This process is biologically important because spacers that are present at the beginning of a locus tend to be transcribed to higher levels, loaded, and the guide RNAs are loaded into more surveillance complexes, which then gives the bacteria more effective immunoprotection against the most recently encountered phage. So the question I had when I started my postdoc is, how does the integrase recognize that this repeat is the first one, provided since this sequence is exactly the same as that one and that one? I hypothesized that there might be upstream DNA motifs that denote that repeat as being the first one. And to tackle this project, I teamed up with a couple graduate students, Murat and Tanner at the time, and an undergraduate, Pusha. This hypothesis had been informed by a series of groundbreaking papers from Jennifer Doudna's lab in a couple years prior to my start that showed that a protein called integration host factor, or IHF, in E. coli bends the leader DNA, or DNA upstream of the CRISPR repeat, into a conformation that stabilizes recruitment of the integrase through the first CRISPR repeat by bringing into contact with the integrase an upstream DNA motif. So the results from our bioinformatics search are shown here. On the y-axis, we have the phylogeny of different CRISPR systems, and on the x-axis, I have position in DNA upstream from the first CRISPR repeat. And so we're going further away from the CRISPR repeat as we go in that direction. For the type 1F system specifically, this analysis revealed the conservation of 4 specific DNA motifs that are conserved not just in their sequence, but they're in their exact position relative to the repeat. 2 of these motifs, I was able to tell just by their sequence, seem to be IHF binding sites. And to provide a little bit of background, IHF had been shown in 1996 to introduce these huge 180-degree bends in the DNA that it binds. And it binds to a very specific sequence motif. The other 2 motifs that we identified seem to be specific to the type 1F subtype, and they occurred as inverted repeats of each other that equidistantly flanked this distal IHF site. And so these motifs are conserved in hundreds of different type 1F CRISPR leaders. And to give an example of what this might look like in a linear piece of DNA, I'm showing here the first bit of the repeat, IHF site, upstream motif, another IHF site, and another upstream motif. And this furthest motif is about 150 or so base pairs away from the repeat. When we expanded our search to a few other CRISPR systems shown here, we found that leaders of these other CRISPR systems have their own unique arrangement of both proximal and distal IHF binding sites, as well as proximal and distal upstream motifs, where the upstream motifs seem to vary between different subtypes and seem to be specific to the given subtype. And so we think that these upstream motifs are co-varying with the Cas1 integrase, or the Cas2. The other surprising finding that came from this analysis is that about 2/3 of the type 1E CRISPR leaders that we studied didn't have any IHF binding sites. They instead had 3 DNA motifs that we couldn't identify matches to in the database. And when we looked back to where these different CRISPR leaders were coming from, which microbial genomes they were coming from, we noticed that all of the CRISPR leaders that had IHF binding sites seemed to come from a phylum of bacteria called Pseudomonadota, which is known to encode the IHF protein. Whereas these type 1E leaders that don't have any IHF binding sites come from a different phylum of bacteria called Actinomycetota, which has IHF-like homologs, but doesn't have the canonical IHF protein. And so this is starting to tell us that maybe there's a co-conservation of these leader motifs, not just to the Cas protein itself, but then also maybe to different host factors or DNA bending proteins that are present in the microbial genome. The motifs that we identified in the CRISPR leaders could have many different roles in CRISPR biology. They could be regulating transcription or integration. So I designed an in vitro assay to test their specific role in regulating integration. And on the top there, I have a scheme of the different components that I mixed together in a test tube, Cas123, IHF, foreign DNA, and then a fragment of the CRISPR locus that has 2 repeats and 2 spacers, and all of the leader motifs that we identified. It's labeled on the 5' ends with P32. If integration of the foreign DNA occurs at the first repeat, at the leader repeat junction, at the repeat spacer junction there, then we'll produce 2 labeled integration products that are fragments of the original DNA. And then we can resolve those integration products on a denaturing polyacrylamide gel. And so for the wild type leader sequence, we have some remaining unintegrated CRISPR substrate. And then we have a leader site integration product and a spacer site integration product that migrate faster through the gel because it's a smaller DNA fragment. If we make mutations to the proximal IHF site or the proximal inverted repeat, then we start to decrease the amount of leader site and spacer site integration that we see, suggesting that these motifs are necessary for facilitating integration. We can then do these experiments for mutants to all 4 of these key motifs and show that relative to the wild type sequence at 1 here, mutating any of these motifs tends to decrease the amount of integration that you get. So at this point, it's shown that the conserved sequences of these motifs was important for integration, but I was also struck during our bioinformatics search at how strongly conserved the exact positions were. And so I hypothesized that either the exact position of these motifs or the placement of these DNA motifs along the helical axis of double-stranded DNA might be important for coordinating or facilitating integration to repeat. So I made a series of insertion and deletion mutants designed to move these motifs. Right, and to remind people, there's about 10 or 11 base pairs that are needed to get a full turn around the helical axis of DNA, as I'm sure many of you know. So I designed a few insertion and deletion mutants that would move motifs in and out of phase with respect to the repeat. Where if we consider the wild type as our 0, then deleting 5 base pairs would move motifs to the other face. They would rotate the motifs about 180 degrees. And I've then also made insertions of 5 base pairs, 10, and then 17 and 20. I then used these leader mutants and performed my integration assays in vitro, and then plotted the integration activity as a function of the total insertion or deletion. So the wild type sequence is here, where we have maximal leader site and spacer site integration activity. If we delete 5 base pairs, then we decrease integration to be close to 0. Similarly, if we add 5 base pairs, we again get very little integration. If we add additional nucleotides and now bring it back up to 10, we're rotating the motifs completely around the helical axis of DNA, bringing it like a full 360-degree around that axis. And so we end up recovering wild type-like integration activity. If you take it out further to 17, then you decrease integration again. And up to 20, you start to increase integration activity again. You can then fit these data points to find the wavelength. And when you do that, you end up with a number that is extremely close to 11 base pairs, suggesting that this behavior might be due to the structure of DNA. And so these experiments suggested that there was something important about the shape or the exact presentation of these motifs in 3D space for regulating integration. And our data is telling us that some of these motifs that are up to 150 or so base pairs away are regulating chemistry at the repeat over here. To actually visualize how that's happening, we had to use structural biology. And so we turned to cryo-EM. And so to set this up, I assembled an integration complex in vitro using purified IHF, purified Cas123, and then I annealed 4 different DNA strands that would end up mimicking a half-set integration intermediate, where one end of the foreign DNA has been integrated at the leader repeat junction there. And then purified this complex by SEC and took thin fractions along the elution curve, and then analyzed these fractions separately on SCS page gels and a urea page gel, and chose a fraction that had all of the protein components and all of the nucleic acid strands that we expect to be present in a fully assembled complex. And I'm sure many of you know more about cryo-EM than I do, but to make sure everyone's up to speed, I thought it'd be fun to walk through the basic process of preparing the sample for cryo-EM and imaging it. So we take a few microliters of our purified complex, apply it to a grid that's the size of a ballpoint pen, wick away most of the sample, and flash freeze it in liquid ethane. Will and Colin helped me prepare the sample on the grids and take in some of our initial images at Montana State University's cryo-EM facility. We had also in the meantime applied for a GUP1 proposal at NC-CAT, and so they helped us collect the larger data set that we ended up using for our model building. The flash freezing of our complex captures it in various orientations in ice. And this grid with our frozen sample is placed into an electron microscope, which shoots high energy electrons at our complex, and we get a transmission pattern of our complex in different orientations on a direct electron detector. And these are some of the micrographs that we got. We ended up collecting about 12,000 movies. And you can see that there's a lot of heterogeneity, both in exactly what orientation we're looking at, and then also of the complex itself compositionally. So you can see that some of the complex is probably starting to fall apart. There's these long strands here that are probably free DNA that have separated from our protein. But there are some still intact complexes, like this little structure right here. At this stage, we can take a blob picking approach to grab some initial particles of interest. And then we use computational approaches to combine these 2D electron shadows into a back-projected 3D density map that represents our complex and what these shadows, 2D images represent. And so I hope that you can appreciate at this stage that what we put into the test tube looks very different from what we got out of our cryo-EM analysis. And I'll walk you through some of the details of those changes in the next few slides. So right into that density map and then build in the atomic model. And on the bottom here, I have schemes of the different components that are present and that we'll touch base on. So the Cas2 domains form a homodimer at the center of the complex, which acts as a nucleus upon which you get the binding of two Cas1 homodimers, where one Cas1 subunit in each homodimer is associating with the Cas2 interface on either side. And so now this is the core Cas1-2 heteroxymeric complex that is present in common to all CRISPR systems that have to adapt to RNA or DNA. Unique to the type 1F system that I was studying here is the fusion of these massive Cas3 helicase nuclease lobes to each Cas2 subunit, which in the integration structure are arranged in this sort of planar orientation with respect to the other subunits. This is very different from the structure that we had previously solved for APO Cas123, which had shown a more compact structure where the Cas3 domains are bunched up against Cas2 and including access to it. And to give you a sense of what's happening, if you morph between the two structures, we see that during integration, the Cas3 lobes are rotating out and away by about 120 degrees. And this rotation frees up new binding sites on Cas2 that we previously didn't know about. In the structure, the foreign DNA fragment is docked onto the underbelly of the complex here, where the 3' OHs are bound in the Cas1 transesterification active sites, ready for integration into the CRISPR repeat. We think that it is this pre-spacer bound or foreign DNA bound version of the Cas53 complex that is searching the cell for genomic DNA that has been pre-bent into a structure that recruits the Cas123 integrase, such that binding of IHF to this distal site here brings these two inverted repeats, which would normally be pointing away from each other in a linear piece of DNA, to be parallel DNA pillars that act as a landing platform for the Cas123 integrase, where each Cas2 subunit makes near identical interactions with a given inverted repeat. And we couldn't find sequence-specific contacts, at least at the resolution we had, between Cas2 and any of the nucleotides in the repeats, in the inverted repeats. Even though we had our biochemistry results showing that if you scramble this sequence, keep the composition the same, but just change the order of the nucleotides, you ablate integration activity. So we hypothesized that the Cas2s are somehow indirectly reading out the shape or the deformability of the DNA in this unique sequence. These Cas2s kink the DNA about 30 degrees when it exits up and away. And so we think then that the role of the proximal IHF binding site is to introduce another 180-degree bend in the DNA that's exiting out to then loop back in the downstream DNA and dock the first few nucleotides of the first CRISPR repeat right at the Cas1 active site here. And so this is how you get specific integration at the first repeat, because this recruitment of the integrase and this docking of the repeat will only occur for the first one in the locus. The other repeats are going to be much further away from the complex at this point, way past the ceiling. And so a surprising thing from the structure was that there's a lot of recognition of DNA motifs and coordination of those DNA motifs that occur upstream of the locus, but we couldn't identify sequence-specific interactions between Cas1 and the repeat, and there didn't seem to be much context happening there. So I hypothesized that maybe the CRISPR leader might be sufficient to promote integration at diverse downstream sequences. And so to test that, I performed a series of experiments where we have our similar integration assay that I showed previously. And I start to mutate this repeat sequence for repeats found in other CRISPR systems. For the type 1F repeat, which is basically the wild type sequence, we have some product for no integration. We have quite a bit of leader side and then some spacer side integration happening. If we mutate the 1F repeat to a 1E repeat, then we retain very similar amounts of leader side and spacer side integration. If we mutate that repeat to a 1C repeat, then we start to decrease the leader side and spacer side integration. And if same thing happens for the 2A system. If we mutate to this repeat sequence, we have a little bit less leader side and spacer side integration as compared to the 1F and the 1E sequences. To give you a sense of how different these repeats are, I have the alignment here. Across these 4 different repeats, the only thing that's conserved is the 2 first nucleotides, a 5' GT. There's no strong sequence conservation within the repeats themselves. They all have different internal palindromic sequences that are used to define CRISPR repeats. They all also have very different lengths and very different GC contents. Right, yeah, so that's there. So based on this initial set of experiments, I wondered whether leader side integration might require a 5' GT, given that all of these substrates seem to be able to undergo that part of the reaction, the leader side integration part. And maybe there seemed to be a loose trend between the length of the repeat and whether spacer side integration was happening. So to test where Cas1 specifically integrates at a 5' GT, dinucleotide performed some additional integration assays. For the wild type 1F repeat, we see robust integration at the leader and the spacer side. If we mutate this 5' GT to an AA, then we completely ablate integration and nothing happens. Conversely, I noticed that the complementary strand at the end of the repeat here, where the second transesterification reaction needs to happen, is not a 5' GT. It is typically in the native sequence a TT. So I wondered whether mutating the top strand to AC, which would then cause a 5' GT to be present on the opposite strand, might increase integration rates or efficiency. And our in vitro biochemistry showed that we get about 2-fold increase in spacer side integration if we recapitulate a similar sequence on the other side of the repeat. And if you combine all three mutations, you don't see any integration happening, which just verifies that integration at any part of the spatial side of the repeat is dependent on leader side integration happening first. It's this two-step process. These data led us to propose this model where we think that the really high levels of IHF present in the cell likely prefold the genome ahead of the CRISPR locus into this S-shaped S shape at any given point in time. The Cas123 complex, when it's free in solution, is in this more compact state, and binding to foreign DNA to the underbelly of the complex causes that rotation of Cas3 lobes out and frees up new DNA binding sites on Cas2, which are then necessary for docking the Cas123 integrase at the genome, where this complex then cooperates with IHF to further fold the leader and dock the first repeat at the Cas1 active site, where the only sequence-specific requirements might be a 5' GT at the beginning and at the end. And that allows you to then get specific integration of new foreign DNA at the first repeats in a given locus. Going forward, my lab will be studying how other microbes use different types of DNA bending proteins and DNA sequences to regulate integration in the context of CRISPR adaptation. We're also interested in how the activities of diverse natural integrase fusions are regulated during this chemical reaction, and understanding how these systems function will allow us to more efficiently repurpose them for Cas-based applications that record DNA or RNA transcription into CRISPR loci. And so some of my new lab members are tackling different projects along this broader aim. We've recently updated this initial bioinformatics I showed you earlier to identify new conserved DNA motifs upstream of many more CRISPR loci. So Rod, graduate student Blake's lab, downloaded all complete bacterial and archaeal genomes from the NCBI database, and we used the software to identify all CRISPR loci in those genomes, and then grabbed all the CRISPR leaders from those loci. We then separated all of those leaders into bins based on taxonomy and on CRISPR subtype. And to take you through very briefly just the 1E data, we found that the DNA motifs that we identify in 1E leaders is influenced by the phylum that leader occurs in. So all the Sumilodota 1E leaders tend to have at minimum these two motifs here, and then sometimes distal copies of them. Those in Actinomycetota have novel motifs, and those in Bacillota again have completely different motifs from those in Actinomycetota. And so this trend where there's coevolution with taxonomy and CRISPR subtype seems to extend to this larger analysis of many more CRISPR systems. And this is now a springboard for our future experiments. We discovered in total 87 different DNA motifs that are strongly conserved in these CRISPR leaders and occur at very specific positions relative to the repeat. 14 of them had been previously shown to have some sort of role in integration, transcription, or RNA processing. But the remaining 73 have unknown biological roles, and we anticipate some of them are playing a role in integration. And I'll tell you very briefly about some new research we're doing for an archaeal CRISPR system. So to give some context, a couple of years ago now, Mike Turns' group, who studies CRISPR immunity in the Archaea Paracoccus furiosus, showed that adaptation in that system seemed to be dependent on histones that are present in Archaea, which was news to me because I didn't know that Archaea had histones until I read this paper. And the way that they discovered this is they isolated chromatin or genomic DNA from the model organism and then performed this nuclease digestion assay. And they saw this very strong banding pattern. If you then sequence the protected regions and map them across the genome, You get several different types of peaks along the entire genome, but what was interesting to them was the strong protection at very specific positions in the leader region for one of the CRISPR loci, suggesting that histones are binding specifically to the CRISPR leader. They then designed this adaptation assay that they used to monitor CRISPR adaptation in the cell. And they showed where primers are amplifying from the leader and from the spacer 1. So if no adaptation occurs, then you have a very short PCR product. If new spacers get added, then now you've increased the distance between the two primers and you have a longer PCR product. You can then resolve these reactions on an agarose gel. And so if you have no Cas1 or Cas2, you have short PCR products indicating no adaptation. If you have histone A, histone B, and Cas1, Cas2, then you get a ton of integration happening. If you mutate histone A or histone B, then you start to decrease the amount of integration that happens. We're interested in determining a structural explanation for how histones are regulating adaptive immunity in this very direct manner. And we've identified conserved DNA motifs that occur up to 500 base pairs away almost in these leaders from the system. So this complex might be very big. And might be challenging to tackle. Ariana and Kat are two undergraduates that joined me last summer, and they helped me perform some of our first integration assays in the lab, where we mixed together histone A, Cas1-2, foreign DNA, and a fragment of DNA meant to recapitulate the CRISPR leader and the first repeat or two of the locus. This organism typically grows in hydrothermal sea vents at close to 100 degrees Celsius. So we perform our integration reactions at around 74 degrees Celsius. And then we resolve the products on a denaturing polyacrylamide gel. If you incubate just the CRISPR locus and the foreign DNA, no integration happens since you have a small band at the bottom. If you add Cas1-2, then you start to see spurious integration events across the entire CRISPR locus. If you add a little bit of histone, where about 10% of the CRISPR should be bound by histone, then you start to see a bit more site-specific integration at the first repeat. And then if you increase the amount of histone to ensure that about 90% of it is bound by histone, then you see much more specific integration at the leader repeat junction spacer side of the first repeat. In closing, we think these types of DNA structures that regulate DNA mobilization aren't unique to CRISPR integration complexes. For example, the motif architecture and the overall shape of DNA in the type 1F CRISPR system looks a lot like what's happening for the lambda phage excision complex, where you have multiple copies of inverted or direct repeats present at the ends of the DNA here, and multiple copies of IHF binding sites that help to organize the DNA substrates and the protein active sites in 3D space in just the right conformation to facilitate either integration or excision of a given DNA sequence in or out of the genome. In both of these cases, DNA seems to be acting as a scaffold that helps to arrange the enzyme sites in 3D space. And as I close, I want to point out that DNA integration underpins some of the most important biological processes in all living organisms, from bacteria to humans. So for example, integration was originally shown to be important for controlling plant phenotypes like corn color, CRISPR-based adaptive immunity, like we talked about today, antibody diversification in eukaryotes, the lysogeny of phages that end up making them more pathogenic, the domestication of retroviruses that then led to the development of what we now know as the placenta, and new types of gene editing devices that rely on CRISPR-associated transposable elements. So I just started my lab at Penn last year, and I hired a bunch of wonderful undergrads, PhD students, and postdocs, and these are my funding, and thank you so much for listening. Yes.
Hold on. There are people online, so let me give you the microphone.
What was the resolution of your first reconstruction that you showed? And do you have a sense of the heterogeneity in your data sets?
Yeah, so the overall resolution was 3.4, I think, angstroms. And some parts, as you might imagine, are higher resolution than others. So like the very core of the complex where the Cas2 is much higher resolution than the arms of the DNA. So those two bends, so this one and this one, they sort of, if you do a 3D variability analysis, they sort of wave at you. So this one goes like this, and then the other one acts as a swing and waves back and forth. In terms of compositional heterogeneity, I did also notice that I got several complexes that were mostly Cas1, 2, 3, and only a bit of the DNA bound. And then as you saw in the micrograph, it did seem like there was maybe some free DNA that was just splayed out linearly in the sample too. Does that help? Yeah.
Victor.
So one aspect, insertion is you insert 20 nucleotide in. So your structure give any insight why and how this is a case?
Oh, okay. Like how the size of the foreign DNA fragment is specified?
Yeah.
So I think some of the integrated structures that came before mine specifically tried to answer this question because it was a core question at the time. Yeah, so the Cas1 subunits have these little wedges that sort of measure out how much duplex DNA can stretch from like one end of the integrase to the other. And then on top of that, there's then just like five nucleotides, nucleotide single-stranded bits of DNA that then have to like thread down into the Cas1 active sites. And so it's specific portions in the Cas1 that really like dictate how much DNA is being bound there and what the rough size is. Some integrases seem to be more stringent in size than others. And so if you like look at spaces that are naturally occurring in CRISPR loci, for certain subtypes, it seems like the integrase is maybe more like a rock. And there's very specifically, oh yeah, you're always integrating 34 base pairs, for example. Whereas others, for many type 3 systems, for whatever reason, there's like a larger spread. And so maybe sometimes even like 10 nucleotides or so.
I enjoyed your talk. I think I actually have a very similar question. So pardon my ignorance and pardon me if you've already answered it. So my question was, so you have to cut both ends of that spacer. Okay. And I imagine then you have to separate the two strands so that you then duplicate the spacer. And so are those cutting sites explain that? You didn't actually have that in your cartoon, but I think it's the next step.
Yeah, right. You're very observant. I didn't have that in my cartoon. I do have a cartoon for that, and it's not in this. Yeah, let me think. I'll go to maybe one of the integration. Yeah. All right, so 1, 3' OH is going to attack this junction. The other one will attack the other junction of the repeat. And I'll answer the first question you had after this one, actually. So what happens is that now it's the repeat that has to be melted, actually. The foreign DNA stays double-stranded. Melting of the repeat causes you to have a single-stranded repeat here, your foreign DNA, and then a single-stranded repeat here. These single strands of the repeat are now templates that are filled in to now reconstitute a full repeat here and a full repeat here. And so that's how you get duplication of the CRISPR repeat and preserve that repeat spacer architecture that's in the CRISPR locus. For your first question of how do you get this fragment to begin with, is that what you had?
I was asking whether the cutting sites were then perfectly positioned on each end of the repeat, the GT, and I guess it would be AA.
Yeah, so yeah, it's a very specific integrase that will integrate like right before the GT. So that 3' OH gets connected to the G on this strand, and this 3' OH gets connected to the in the wild type sequence, the A on this strand. This is,
Yeah, so is that space in your structure or is that the next step?
oh, yeah, I only captured this first reaction in the structure. Yes. Sorry for misunderstanding.
That's my question.
Yeah, okay, perfect. Yeah. So I only captured that first step. And really, I didn't provide the metals to allow the complex to perform the other integration reaction in this project. We have done that experiment. The data for that is a lot more noisy and a lot more difficult to sort through. But Will, who's working on with me on that project, has made some progress recently. But yeah, if you provide the metals, then this repeat will stretch out and, well, not stretch out. There's 2/3 of it are not resolvable in our structure. They're floppy like this, and the density just peters out after here, really. But it should integrate over there. Yeah.
Nice talk, Andrew. I have two questions. One question about this dramatic Cas3 rotation. What triggers this rotation?
The rotations.
Cas3.
Oh, the Cas3 rotation. Yeah. Okay. So we think it's binding at the pre-spacer, and one reason why I think that is there's a disordered linker that typically holds the Cas2 subunit to Cas3. And in the apo structure, that linker is in a certain conformation. But when the pre-spacer binds, it would clash with where that linker is typically partially ordered in the apo structure. And it seems to actually like press that linker flat. And I think we think it's forcing it into a slightly different conformation that then cascades into rotation of the Cas3s outwards, which seems to be a slightly more stable conformation when that linker is no longer allowed to be in its initial position.
And my second question about orientation of loading of Cas1, Cas2, Cas3 to face distal end to leader repeat junction.
Yeah, okay. I wasn't able to answer that with this structure, so I actually don't know. Well, this integrase is very specific in that if we give it a pre-spacer that has a PAM on it, it won't integrate that. It'll do the correct orientation for integration. So it'll integrate the non-PAM end into the first the leader repeat junction, and then the process just stops there, presumably because we're not including whatever nuclease might be needed to trim the PAM, and then cause integration at the other end. I don't believe I have the PAM in this structure, so I can't say if there are specific contacts between Cas1 and the PAM that are reading that out in this structure. Although I think like in the 1E structures, that seems to be the hypothesis there. There's structural data that the PAM is read out by Cas1.
Greg always sits as far from the speaker as he possibly can.
And so I need the microphone anyway. Thank you for the talk. I have two questions also. One, you introduced insertions 5, 10, 15, well, 17, 21 base pairs. I noticed you have more than one IHF site. What would happen if you introduced 5 insertions in one and 5 in the other?
Yeah.
Is there any crosstalk between them?
Let me think. So like if we insert 5 at this, so this is repeats, this is the first IHF. If we insert 5 here, then maybe that flips this bend to now like point that way. So really this IHF site, if you insert 5 here or here, that's now twisting. It does a few things, but it definitely twists where these inverted repeats are relative to Cas1, relative to Cas2, sorry. And so that would mess up which face of the inverted repeat is being presented to Cas2. So you'd probably mess up interactions between Cas2 and the inverted repeat if you're making mutations here. For mutations that occur, or insertions, deletions that occur around the IHF proximal site, I think you're causing like really large rotations of like where the other bits of the DNA are being placed in 3D space. It's like this whole thing is going to swing around this axis if you make insertions or deletions here.
The other question's a really short one. You were mutating the 1F to say 1C1A. There's a band above your leader sequence on the gel. What does that correspond to?
That's great. It's a non-specific integration product, and I don't know the exact identity of it. We would have to sequence the reactions probably to determine exactly where that is. But thanks.
Great talk, Andrew. I just had a question. You had also shown some sequences that didn't have IHF binding sites. Do you think in those systems there's some other mechanism for bending the DNA, or is somehow the Cas1, Cas2 complex more adapted for, I don't know, bringing those different pieces of DNA together?
Yeah, absolutely. Yeah, that's a great question. So, as you might know, I think there's several examples that have been studied by either Jennifer Doudna's lab and/or Alon Koh's lab of some Cas1-2 integrases that seem to somehow independently recognize the repeat without the need for additional DNA that's really far upstream or DNA bending proteins. But we think that given the bioinformatic analysis that we've done, that we have a hypothesis that in those other bacteria, there are other DNA bending proteins that are facilitating a similar type of structural rearrangement that promotes this reaction. And so IHF is limited to Pseudomonodota, but there are other type 2 DNA binding proteins in other phyla that might be doing that. And then there's evidence in Archaea, which don't have IHF, that histones might be playing a similar-ish analogous role.
Thanks.
Thanks.
Let me just check the chat. Okay. Could you say something about either other known roles or other potential roles for IHF in bacterial physiology?
Yeah, absolutely. That's a great question. So IHF was first discovered and first given that name because it's integration host factor for lambda-phage. So in that instance, it's an interesting dichotomy because in that instance, lambda is repurposing this core protein involved in E. coli physiology for its own purposes to integrate into the E. coli genome. Whereas here we're showing how the CRISPR immune system is adapting to phages like lambda-phage with the help of IHF. So IHF doesn't just regulate integration processes like lambda-phage or CRISPR. It's also involved in regulating transcriptional events where it does a similar thing. It bends DNA into these U-bend shapes to recruit like upstream DNA sequences that stabilize the transcriptional complex and allow it to then kick off a successful transcriptional event. And there's also roles for it in packaging and the genome in bacteria too.
Right. So this is an example of that which makes you can also break you.
Yeah, right.
Terrific. Okay, well, we're just coming up to the hour. Thank you very much. There's about 40 people in the room and another 20 odd online. You've attracted a great crowd for us today. Terrific talk.
Thank you so much for inviting us.
Great to be able to host you here at Rutgers. Thank you very much, everybody. See you in 2 weeks.